My presentation is about the presence of hyaluronan in fascia and how it changes with aging or in other condition. First of all, when we speak about fascia, we need to understand that the fascia have a double component. They have a fibrotic component and the loose connective tissue. The fibrotic component allow the mechanical transfer, so it work like a tendon connecting different part of the body, transmitting the force, organizing the movement, but then we have the loose connective tissue, and the loose connective tissue is the element that allow the fascia to glide respect to the underlying muscle, but allow also the nerve to glide respect to the fascia, allow at each fascia to glide one respect to the other. So we need both the component to have a good facial system, a very well organized fibrotic component, and a good gliding to allow the sharing motility. And what permit the gliding? We published still in 2011 the first paper about the hyaluronan, and we highlight that the fascia are rich in hyaluronan. And we were also able to understand, almost 10 years later, the cell that are able to produce the hyaluronan. So in the fascia, we have not just the fibroblast, but we have another type of cell that we call the fasciacytes, that are specific cells that are able to produce the hyaluronan. Here you can see the cell that have a round shape, are in the middle of the fascia, but above all are organized as a small cluster in the surface of the fascia. And we stain that cell with the Alcian blue, with the HIBP, and we see that all around they have like a ring of hyaluronan. We analyze them also with the electronic microscopy, and you can see as in the extracellular matrix of the fasciacytes, we have a lot of hyaluronan. On the contrary, the fibroblast produce above all collagen fiber, elastic fiber. So probably they derive from the same progenitor, but then according with the location, the mechanical input, or I don't know what could be, they can divide in two different type of cell, one more devoted to the production of hyaluronan and the other of fiber, collagen and elastic fiber. Here you can see a cluster, a typical cluster with 4 cells. You can see that are just in the surface of the fascia, and probably during the motility, the sharing motility, they are stimulated to produce hyaluronan. We try to analyze and to compare with the facial fibroblast to understand if they are the same cell or not. But you can see that there are some elements that can differentiate the fibroblast respect to the fasciacytes. So there are many aspects in common, but not all. Besides, we try also the stain of collagen type 2 because we had the doubt that are like a metaplasia of the fibroblast, but they are collagen 2 negative, so they are not chondrocytes or they are not in a phase of metaplasia respect with the chondrocytes. And we are not the only person that found the fasciacytes, but still in 1933, there was this paper that published this line of hyaluronic acid secreting cell in the wrist retinacula. So in the surface of the retinacula of the wrist, they show this line of cell that are able to produce hyaluronan. Again, are in a very important gliding surface of the fascia, because in the retinacula we have a lot of movement, and consequently we have a very well organized line of cell. So we can affirm that we have estimated that in healthy fascia, almost 30% of the fibroblasts are really fasciacytes, so they are devoted to produce hyaluronan and not extra fibrous cell. We were also able then to quantify the amount of hyaluronan in the different type of fascia. And here you can see again that the amount of hyaluronan is very specific for the different type of cell, the different type of fascia, and is defined by the gliding ability of the different fascia. Because where we have an epimysial fascia, so a fascia that is totally attached to the muscle, we have a very small amount of hyaluronan. In the fascia lata or in the rectus sheath, where we have a pretty good gliding, we have around 30-40 microns, and that quantity was confirmed also by many other studies later. In the retinaculum, where we have a lot of movement, where we have the tendon under that need to glide, we have 3 times more hyaluronan. And we were surprised that that quantity is pretty constant in all the different persons. Pretty constant, but here I have a video, but unfortunately I'm not able to share you. But with the ultrasound, and maybe Carmelo then can show you better, we can analyze the ability to glide of the different fascia. But then we, with such type of quantity in healthy people, we analyze the amount of collagen and hyaluronan in other type of fascia. And in this study was analyzed the fascia lata in a patient with hip osteoarthritis. Are people pretty aged, around 80 years old, but you can see that we have much more collagen type 1. The collagen type 3, we have some doubt about the result. And surely we have a huge decrease of the hyaluronan. So it means we know that in patients with osteoarthritis, we have a decrease of the production of hyaluronan inside the joint. But this is the first study that suggests that the decrease of production is also outside the joint, in the fascia. And I'm surprised with respect to your study because we found an increased amount of collagen type 1. Also in aging, we found a similar aspect. Again, a huge decrease of hyaluronan, and here I have not reported the study of collagen, but we found an increase of collagen type of 1. But we did a similar study in rats to compare different ages. And what we can understand, this is around 11 years old, 35 years old in human, and 70 years old in human. And the decrease of hyaluronan is not constant in our life. But it seems that when we are young, we lost much more hyaluronan. Then we reach a more or less stable situation, and then the decrease is much slower. We have now also another study that analyze the fetuses and just until the delivery, and we have much more hyaluronan. So it seems that we born with fascia that are very rich of hyaluronan, with a few collagen fiber, not very well organized, and then with the movement, we organize our fascia, we create a more stable condition, we decrease the hyaluronan, and around 20, 30, we don't know exactly the age, we reach a more stable situation in our fascia. It means that we lost in ability to move because we have less hyaluronan, but we have more collagen fiber, very well organized. So we have a good force transmission, good coordination among the muscle, and so on. In that situation, we can maintain more or less the situation for a long time. And only if we have a pathological condition, we have a further damage of loss of hyaluronan. Also, in the plantar fascia, we found a very strange situation because in the majority of the patient, we found around 40 microns, exactly as the fascia lata, the rectus sheath, that is pretty similar, are fascia that are pretty similar. But in a male pretty young, we found 3-4 times more hyaluronan. Is the most, the biggest amount that we found. And in a patient with fibromatosis, much, much less. So it means that the pathology, for example, the fibromatosis, can affect the hyaluronan and the ability of the fascia to glide. But honestly, I don't know why we have so high level of hyaluronan in such patient. Another aspect that we need to consider if the possibility to stimulate the production of hyaluronan. And we were curious about the endocannabinoid system because we were able to found the receptor of the endocannabinoid system in our fascia. So we isolate the cell of from the fascia, and in the lab we stimulate the fascia with an agonist of CB2. And we were surprised to see that the cell activate and start to produce hyaluronan in the extracellular matrix. So it means again that the endocannabinoid system is able to improve the production of hyaluronan in our fascia. And here you have the different stain. You can really see here. This is HBP, and you can see the cell that is releasing the hyaluronan in the extracellular matrix. But we try to understand also if the mechanical input can stimulate the production of hyaluronan. And to give a very precise input, we use the focal shock wave, a very low level, and we stimulate the same cell culture with the shock wave. And we demonstrate that just after few hours, we have a good release of hyaluronan in the extracellular matrix. So the effect of the shock wave, the focal shock wave, is again to stimulate the production of hyaluronan in our fascia. And to understand what is the element that can connect the mechanical input to the production of hyaluronan, we check the proteins that are able to do the mechanotransduction. And in specific, we check the YAP protein. We know that the YAP protein is one of the most important protein that is able to do mechanotransduction. We found the YAP protein in our fascia. So we did the stimulation with shock wave, and we see that the treated cell increase the expression of YAP protein. And again, we have the production of hyaluronan in the extracellular matrix. Then we decide, you can see also the increase of the production of the YAP. We have also the release of collagen fiber, but only just after 2 hours, and then is not maintained that element. But to confirm that really the YAP is the element that is able to connect the mechanical input to the release of hyaluronan, we block the YAP protein with the verteportil that is an element that block that receptor. After this treatment, the same treatment with shockwave was not able to release hyaluronan in the extracellular matrix. So to conclude, we can affirm that the fascia is a very complex structure. More I'm studying the fascia, more I think that there are many different aspects that can affect the fascia. Surely the mechanical input due to the shock wave, but also the movement can affect the fascia and can increase the gliding. The aging and the life can change the hyaluronan. The arthritis can affect the hyaluronan, and also probably the hormones. We have tested only the endocannabinoid receptor, but we have in mind to test other type of hormone to understand really if the different element can affect and can regulate up and down the production of hyaluronan. Thank you for your attention.